Hi, my name is Erin, and this is my protein poster. The protein I'm talking about today is hexokinase, which is also known as ATP dehexose 6 phosphotransferase. It's found in all living organisms from humans and animals, bacteria, fungi, and plants. Inside the cell, it resides in the cytosol, where it catalyzes the first committed step of glycolysis, the phosphorylation of glucose. It belongs to the transferase enzyme class with EC number 2711, and it's a high affinity for glucose, with a Km value of approximately 0.1 millimolar. Hexokinase works through a well-defined mechanism. The enzyme itself forms the active site, which binds both glucose and ATP. Glucose is a substrate and is phosphorylated at the active site, while ATP donates the phosphate group. This reaction converts glucose into glucose 6 phosphate, also known as G6P, effectively starting glycolysis. Magnesium ions are essential cofactors, stabilizing ATP during the reaction. The total number of amino acids within hexokinase is approximately 917. Its unique features consist of two connected domains, one for catalysis, which is the C-terminal, positive, and one for regulation, N-terminal, negative. It's controlled by feedback inhibition of G6P and can form dimers of higher order structures that affect activity. As previously mentioned, the cofactors include magnesium 2+. The proportion of alpha helices, hexokinase is composed of a mix of alpha helix and beta sheets, roughly 40% of them being alpha helices. Number of disulfide bonds. There are no disulfide bonds confirmed or evident, so there is no location. However, yes, there's presence of salt bridges, and we do not have a definitive count. Interactions between positive and negative charge residues, positive being lysine and arginine, negative being aspartate and glutamate. Non-covalent, weaker than disulfide bonds, but strong enough to stabilize protein structures. There is only one subunit, and the type is a monomer. The significance is the quaternary structure of enzymes, such as dimerization of Cluveromyces lactase hexokinase, also known as KIHXK1, regulates activity. The substrate of product binding shifts the enzyme between monomeric and dimeric states, affecting its catalytic function. As you mentioned previously, it takes glucose to glucose 6 phosphate, and its physiological significance is hexokinase exists as different isozymes in different tissues. Isozymes are encoded by different genes but catalyze the same reaction, but can also respond differently to allosteric effectors. It allows tissue-specific control of glycolysis. Some examples of disease conditions, if dysfunctional, can occur would be non-spherocytic hemolytic anemia due to hexokinase deficiency, which is a blood disorder where red blood cells are destroyed faster than they are being replaced. And another example would be cancer. For exhibition allosteric modulators, known inhibition types, inhibitor being glucose 6 phosphate, G6P, inhibition type being non-competitive. Known allosteric modulators is also G6P, type being heterotropic. Binding site of inhibitor modulator is the N-terminal half of hexokinase, which indirectly displaces ATP from active site. And these would be my references page. Thank you.